Welcome everyone to today's event, Promising RSV Vaccines on the Horizon. This event is organized by Science Magazine in collaboration with the Texas Science Festival at the University of Texas at Austin, and I'm Megan Phelan, Director of the Press Package at the Science family of journals. This is a very timely discussion on RSV, or respiratory syncytial virus. RSV, as many of you know, is a seasonal illness that can be deadly for babies and elderly people. And unfortunately, RSV cases surged this past winter, overwhelming hospitals all over the country. Though there's been no vaccine available for RSV for decades, scientists did try one out in the 60s, but it didn't work, and we'll talk about that today. Now, here in 2023, we're in a very different place. Several promising RSV vaccine candidates that work in a whole new way compared to past candidates could be available very soon. In fact, yesterday, one RSV vaccine candidate for the elderly was the subject of review by an FDA independent advisory committee. These advisors recommended that the FDA approve this vaccine. Today, they're weighing in on a second RSV vaccine from another pharmaceutical company, also for the elderly. And we'll have updates there as soon as an hour from now, potentially. Our guests today, including 3 scientists and 1 journalist, are the perfect people to be talking to us about the history behind these vaccines' development and the science it took to get us to where we are today. Before I introduce them, I want to invite viewers to post your questions in the chat. I'd also encourage you to let us know where you're listening from. We'd love to hear where people are joining us from around the world. The discussion today should last about 30 minutes. I have several questions for our speakers, but I also look forward to taking yours in the audience. And now we'll get right to our speakers. So our first guest is Barney Graham. He's a senior advisor for global health equity at Morehouse School of Medicine in Atlanta, Georgia. He spent 20 years as an investigator at the National Institute of Allergy and Infectious Diseases and retired as deputy director of the Vaccine Research Center there. He's received multiple awards in his career, including for his work on vaccines against COVID-19 and RSV. Hi, Barney. It's really exciting to talk to you this week.
It's nice to see everyone. Nice to be here.
Jason McLellan works at the University of Texas at Austin, where the Texas Science Festival is happening now. His lab is focused on understanding and visualizing how hosts and pathogens interact, and he and his team use this information to develop vaccines and immunotherapies. McLellan developed technology that features in most current COVID-19 vaccines, as well as the new vaccines against RSV. Hey, Jason, it's good to be with you on Festival Week.
Hey, Megan, thanks for having me.
Man Chen is a recently retired staff scientist at the Vaccine Research Center, where she worked with Barney and other colleagues. She joined the Vaccine Research Center in 2002. Today, we're going to hear about work she did, including involving international collaboration to support RSV vaccine development. Hi, Man. I can't wait to hear your insights.
Hi, Megan. Thank you. I'm looking forward to talk to you. Thank you. Hi, everyone.
And our last speaker is Gregory Zuckerman, a special writer at the Wall Street Journal, where he's worked for nearly 3 decades. He's also the author of multiple best-selling books. His latest, A Shot to Save the World, the inside story of the life or death race for a COVID-19 vaccine, is reportedly informing an upcoming HBO documentary. It does a tremendous job explaining the technologies behind vaccine development and the heroes, including those on this panel, involved in the process as related to COVID-19. Hi, Greg. How are you?
Hey, everybody. Nice to see everyone.
And I'd like to start with Greg as we kick off our discussion today. And again, people watching, please feel free to post your questions. We'd really like to hear your questions about the science and the technology involved in developing these vaccines and the history of that work, and broader questions too. So Greg, let's start big picture here. Your book about the development of the COVID-19 vaccines talks about how scientists turned years of underappreciated work into life-saving vaccines at incredible speed we'd never seen before. And they did this so quickly in part because they were able to take advantage of insights from RSV vaccine development. So to give our viewers a sense of how these 2 major efforts aligned and diverged, what are the parallels in the way the COVID and RSV vaccines were developed? What are the major differences?
Sure. I'm going to answer that question as an outsider. I'm a journalist. I write about the scientists, the researchers, and the executives. And what struck me writing my book is I kept hearing about the work done with RSV. I kept hearing about the work done with HIV. And I don't think people are appreciative of the persistence and the resilience and the patience that's required for some of these breakthroughs, including RSV. And we're going to hear in a few minutes from Barney and from Jason, and they're heroes. And I think people need to appreciate the level of patience and persistence that's required. And that's in common for both and for all breakthroughs. And also the importance of just basic science, basic research that goes on in labs around the country and around the world for years, people persisting in deep in these labs. And I come from a world where we want results. We want to produce something right away. We want to see it right away. If not immediately, then the next day. And I really have so much newfound respect for these researchers who spend years, maybe even decades, as we're going to hear in a few minutes, on RSV before they get their breakthroughs and the results. And we still need that, and more than ever. And we are potentially on the cusp of a remarkable breakthrough with RSV, where maybe the lives that will be saved and will be helped might even be more so than the COVID vaccines. And not enough, I think, acclaim has accrued to these researchers. So I'm eager to hear about them myself. But there are a lot of similarities in that persistence and in the importance of just basic science. And it's a good reminder for us all of the importance of both.
Yeah, thanks, Greg. I feel so lucky to have these researchers on with us, the way they've persisted, the history they have here. Let's go to Barney on that note. Barney, your history with RSV is long. As Greg alluded to, it's a great story of how basic research puts us in a position to make usable products. Could you tell us how you got interested in studying RSV?
Well, all my work in virology really stemmed from being in training during the advent of the HIV pandemic. So seeing one of the first patients in Tennessee with HIV in 1982 led me to want to do virology. And most virology at that time, in laboratory work at least, was done in the pediatric department. So when I went to Peter Wright and Kathy Edwards and David Carzon at Vanderbilt in their pediatric group, they allowed me to come in as an internist and start work on a respiratory virus, respiratory syncytial virus.
Okay. And so the audience can know a little bit about the history of vaccines for respiratory syncytial virus. There was a trial of an RSV vaccine candidate in the 60s, as I alluded to in my opening remarks, but it led to not an ideal outcome. Can you tell us a little bit about what happened and why, Barney?
Yes, I got into RSV research to study safety, to study vaccine safety, and safety is really the first step. It's do no harm is one of the anthems of medical scientists. And in the 1960s, shortly after Salk had such success with the polio vaccine by just simply inactivating the virus and using that as the vaccine, when RSV was discovered in 1956, similar steps were taken. And by 1965 or so, a whole inactivated virus vaccine was tested in infants, and in the youngest cohort of infants, in a relatively small trial, 31 infants received the vaccine, and 20 of them were infected during that next winter season, and 16 of them out of 20, 80%, had to be hospitalized, and 2 died. And that was a vaccine-enhanced disease. And I spent the first 20 years of my career studying the basis and the pathogenesis and the immunology of the vaccine-enhanced disease, hoping to get to a place where we could safely develop RSV vaccines.
I'm sitting here thinking about this as a parent of a 6-month-old. Very, very concerning results. And so after that first failure in the 1960s, what happened to the pace of vaccine development for RSV, and how did things look? Who was involved for those decades?
Well, that put a stop to RSV vaccine development for a few decades. And then when work was restarted, it was focused in part on the elderly, because what was known is that once you'd been infected with RSV, or once you'd gotten a live virus vaccine with RSV, that vaccine enhanced disease was no longer an issue. It was something that happened in babies if they got that kind of vaccine on their very first exposure to the virus. And so work had started again in adults and by the mid-90s, and in children using live attenuated vaccine approaches. And but it didn't go very far. And so in addition to vaccine safety, you also have to design a vaccine that can work, that can actually stimulate sufficient immunity to be protective. And so safety's first, efficacy development is next. And it took us a few years to get to that. And we did a lot of work with vaccines that elicited T-cell based immunity. And then eventually around 2008 or 9, when Jason came to the VRC, he and I, and he was in Peter Kwong's lab at the time, we started working on a new approach to RSV focused on generating better antibody responses.
That's a great segue. Thank you, Barney. I want to go now to 2013, a few years after the time that Jason came to the VRC. So that year, Barney, Jason, and Man published a paper in Science highlighting research that focused on the new design that Barney alluded to, research that would lay the groundwork for creating the RSV vaccines in clinical trials today. We'll post a link to that 2013 Science paper in the comments. But the paper showed that they developed a candidate that led to much higher protective responses than other leading RSV vaccine candidates. They did so using new insights into the structure of what's called the virus's F protein, which the virus uses to dock to cells. Normally, the F protein flips back and forth, changing shapes after it attaches to a cell. But this group figured out how to freeze the protein into the shape that it takes before it fuses. That's what's called its pre-fusion form. And very important for vaccine development, they showed that a vaccine against RSV would have a greater chance of success in triggering strong antibodies if it was directed at this pre-fusion form of the F protein. But figuring out the pre-fusion structure of the F protein was not easy. Jason, this is your world. Can you explain a little bit about what it took to do this?
Yeah, so the RSV F protein, it's similar to the coronavirus spike. They're fusion proteins. They, as you mentioned, start off in an initial pre-fusion state, fold into a post-fusion state, and that helps fuse the viral membrane with the host cell membrane. That initial state is unstable, sort of metastable, but that's the form we want to use as a vaccine antigen. We want to train our immune systems to recognize this initial form. And so one of the tricks then is, how do you stabilize it in this unstable form? And we want to do that with structural biology, be able to determine high resolution, three-dimensional atomic structures of this protein. So that way we can then rationally engineer in substitutions to help lock the protein. But first we had to get the structure. And to do that, we were able to take advantage and identify antibodies that could bind to the unstable form and lock it and prevent it from switching conformations. And so the first structure we obtained was an antibody-bound complex of the pre-fusion F protein.
Thanks, Jason. Yeah, and now I'd like to talk a little bit to Man. Man, you traveled to China to support this work, and you were looking for the antibodies that Jason alluded to that would bind to pre-fusion F. Can you explain how many antibodies you tested there and how long it took to find those that had really strong protective responses but only bound to pre-fusion F and not post-fusion F?
Sure. First, I want to talk briefly about Jose Malero, who is really important part of this story, who was a virologist at National Center for Microbiology in Spain. Jose Maloro's group found a human serum with high titer of neutralizing antibody retained most of neutralization activity after post-F absorption, which suggested the potent neutralizing antibody was from pre-fusion F. And pre-fusion F would be better immunogen than post-fusion F. In 2011, we started working on solving the pre-fusion F structure because of Jose's work. We knew we need a specific antibody to bind the pre-fusion F and lock it in pre-fusion conformation for crystallization because, as Jason talked, the pre-fusion F is metastable. It's hard to get the crystal of pre-fusion F. We decided to collaborate with Professor Lin Shaoxia at Xiamen University, China, for making the antibody because his group had expertise on making antibody by hybridoma technology. In 2011, I made a trip to Xiamen University. In Professor Xia's lab, Zizhen Zheng and Min Zhao worked on this project. We isolated one pre-fusion antibody, and which was screened out from the over 10,000 clones. We named the antibody 5C4. Then we use 5C4 for the crystallization pre-fusion F protein.
Thanks, Man. So you talked about screening thousands of clones. What criteria did you use to isolate the specific pre-fusion F antibody that you ultimately kind of found and landed on as important to continue to study?
Yeah, we set two criteria. First, the antibody could neutralize RSV, but it should not bind to post-fusion F. By using this criteria, we screen out pre-fusion F antibody.
Thank you, Man. Okay, so back to today. So now we are in an important time here. Several major pharmaceutical companies have used the technique you all have described to develop vaccines that have proved their worth in clinical trials. And one design is a vaccine for people over 60. Another is for pregnant women who could pass antibodies or pass immunity to their babies. There's also a monoclonal antibody, a one-time injection that's being evaluated. That would be for infants. This is tremendous progress. And we've also seen, as I mentioned in my opening remarks, the FDA advisory committees weighing in this week with votes of approval. At least yesterday, we're waiting to see what will happen in the second vote today. But based on the news yesterday and on the vaccine for the elderly to start, Barney, to come back to you, what do you think about when we could expect the vaccine for the elderly, for example, to be approved and available? What might be a possible timeline?
Well, most of these products have received breakthrough status and rapid review privileges. And so once the biologics license application is submitted to the FDA, they have 6 months to give them back an answer. And so some of these products, like the two products that had been tested in the elderly, are under the review today of the VRBPAC committee, the Vaccines-Related Biologics Advisory Committee. And so they'll be voting, they'll be looking at both efficacy and side effects from these vaccines. And then the FDA will have to make a decision within the next few weeks or months. So decisions will be made about all these products by next winter season.
Okay. I want to pause for a moment. I'm sure that that's very interesting for the audience. Audience, if you have questions, please do enter them in the chat. We'll be happy to raise your questions to our speakers. We've talked a lot about the vaccines, but what about the monoclonal antibody? What's its status in review? And when might we and could you explain a little bit about who it's intended for? This is for babies before they would become ill. Is that correct? Jason, maybe we could go to you for this question.
I'll let Barney handle the question about the review in different stages, but in terms of how the antibody works, so it's an antibody we actually used it's a modified form of the antibody we used to solve the original structure. Back then it was called D25, and it binds the apex straight down almost at the top of the RSV fusion protein, and it binds to regions of the protein that need to refold and undergo a conformational change to adopt the post-fusion state. So by binding, really just locks the protein. Now the virus can't enter cells. That's the type of antibody, one of them that we're sort of hoping the vaccines will elicit. That's why we want to use the pre-fusion stabilized form of the F protein, so that way when injected, you can elicit this type of antibody. And so you can do active vaccination, where you inject a viral protein or pathogenic protein and try to have the body elicit those antibodies, or you can do passive immunization and just directly inject the purified antibody. And so that's what this is being developed for perhaps high-risk infants. Maybe they're too young to vaccinate directly. So you can inject them with the antibody and perhaps protect them for the first 6 months of life or the first RSV season.
As a parent with an infant, that's very exciting news. And what about the review status for that one, Barney, the monoclonal antibody nirsevimab?
It's nirsevimab, and it's already been approved by the EMA, the European Medicine Council. So, and it's under review by the FDA. So it should also be reviewed and decided on by next winter season. And yeah, this antibody is derived from one of the antibodies we use to do the crystallization of the prefusion F. And it is given to infants at the time of birth. And it's been tested in full-term infants. One of the things we know about RSV is that if you can give an antibody that neutralizes the virus early in life, it can be done safely and effectively, because there's an antibody called palivizumab that's been used since the late 90s. It's been restricted very strictly to very young infants, 29 weeks gestation or less. But this new antibody that's also been modified with a long half-life mutation, it is being given to full-term infants. So it would be available for all babies born. And has been shown to be highly effective, at least through 150 days.
Very exciting. Okay, so a second monoclonal antibody available for a wider group of infants, not just premature infants. I want to move to a question from our audience. This is on the topic of safety, which you've addressed, Barney, already in response to several questions. And this viewer is asking, what is the correlation between myocarditis and RSV vaccine's role, and what is RSV vaccine's role in recent cases? And what are safety regulations that have been used in the context of measuring these incidences of myocarditis? I think that's the question.
Okay, well, this person's asking about myocarditis. As far as I know, there hasn't been any events of myocarditis with any of the RSV vaccines or antibodies tested to date.
Maybe, okay, so maybe they're thinking about COVID-19. And if the person who posed that question wants to follow up and ask and clarify, please feel free if there's part of your question that I've missed. It looks like we have another question coming in on the chat. So I will wait for that one. Okay. Can you talk a little bit about how RSV and COVID differ in severity for people of different age groups? It's kind of a broad question of comparing the two.
Well, they're different types of viruses, but in some ways they share features. So they're both envelope viruses, they're both RNA viruses, they both have class 1 fusion proteins. But a big difference with RSV is that everybody on Earth has been infected with RSV, and they get repeatedly infected throughout life. And so the severe times for disease are very early in life, before 6 months of age, peaking at around 2 months of age, and then very late in life when you've become frail or you have an immunocompromised state or different conditions that could make the virus worse. Coronavirus, the new coronavirus, one associated with COVID-19, came to the human population for the first time, and there was no pre-existing immunity. No one had ever been infected with this virus before. And so we are now gradually coming out of the pandemic. This virus is becoming an endemic virus, and it will settle into what I think will be a seasonal pattern. It will be coming back around probably most winter seasons, and we'll have to see what kind of disease pattern it eventually assumes. It doesn't appear to cause severe disease in very young infants like RSV, but it will probably always have some severe disease in very old, frail, immunocompromised people.
Thanks, Barney. And now a question for you, Greg. Viewers are wondering if you could talk a little bit about how people have reacted to your book, maybe surprising responses they've had about efforts to make the vaccines, which would also be relevant for RSV.
Well, one of the themes is that those who develop these vaccines are not who you would have expected. You would have expected it to be big pharma, right? And I frankly came into my project a little bit skeptical of researchers, of big pharma. We've written enough stories at the Wall Street Journal and elsewhere about pricing and other kind of issues. And I gained a whole new respect. And I came at this, again, as a skeptical kind of journalist. But the researchers, both on this panel and elsewhere, are dedicated. They're Republicans, they're Democrats, they're not political. And frankly, they're trying to help us all. And in this environment, we're in an environment where, for whatever reason, everything's politicized. It's sad because I kind of play down the middle, and I talk to these people, and they've dedicated their lives to this research, and they have this resilience and this persistence, which is admirable. And frankly, we all talk about technological innovation coming out of Silicon Valley, everything's Silicon Valley, and today's AI. But these researchers, they are at the heart of technological innovation that is admirable, and I think we can learn from, whether we're in this industry or not, and we should be grateful as well. I mean, the reason why we're out and about and doing what we are nowadays is thanks to vaccine researchers like Barney and Jason and others. So I'm not here to defend them, I'm not here to promote them in any way, and I came into this project, my book project, a little bit skeptical. And you talk about reactions, yeah, a lot of people have been surprised about who it was and what kinds of people were responsible for these vaccines, and they were surprised that it wasn't, Merck and GSK and the vaccine giants. So there's a lot of dedication in this industry, and I gained a newfound respect for the researchers.
Yeah, humanizing the story of the work, and I'm emphasizing the gratitude for all of you. And we have a question that relates to the fact that, again, 2 of the vaccines are being reviewed by advisory committees to the FDA this week. Someone's asking about concerns they'd seen about the very small number of cases of Guillain-Barré syndrome, a neurological syndrome. How worried should we be about that? I think they reported 1 case for one of the vaccines for the elderly and perhaps 2 for the other. So very small number, but we did hear those results. Barney, I think this is for you.
Well, this is what I meant about safety first. And the first thing every group developing a vaccine does is make sure the trial is large enough to assess safety, even things that happen rarely. And so there were a couple of what I would classify as demyelinating type syndromes, that either the central nervous system or peripheral nervous system, that were associated or closely occurred close to the vaccine. And And so this is the kind of thing that is such a rare event. It's hard to know how to judge that relative to how many people the vaccine is helping. And it's often the kind of thing that you continue to study what's called post-marketing. So once the vaccine is into millions of people, you can get a better sense. And this would be done in real time. And so if there were events that were concerning, both the manufacturers and the FDA and the CDC would all have access to that real-time data and be able to stop if there were something that looked concerning.
Thanks, Barney. That's really helpful to know how the process would go. Everyone, we are nearly at time, so I want to end with a question for everybody, and I think I'll start with you, Jason. What lessons do you want viewers to take from the history, the story that you've told today, even in short form, about the history of developing RSV vaccines?
I think maybe one thing that's not appreciated is just that the work takes hundreds or thousands of people over decades. So, if you're seeing a few of us, we've mentioned a few names, but it's really just a tremendous number of people dating back to when the virus was first discovered in the mid 1950s. It's a lot of failure. Science takes time. It's self-correcting. Hopefully we ultimately get there and can one day produce effective vaccines. So I think it's exciting for the field to finally have efficacious vaccines being evaluated for approval now.
Thanks, Jason, and for everything you're doing in your lab. Man, how about you? What lessons should we take from this story?
Yeah, I think I have one point. The scientific collaboration is important and improve our research. We collaborated with Lin Shaoxia's group, find the monoclonal antibody prefusion antibody successfully. We were from different research background with special expertise. We were learning each other, sharing the knowledge, expertise, and technology, which speed up the work and improve the efficiency and creativity of the project. And collaboration benefits to all of us.
Yeah, you use the word creativity, thinking about collaboration informing creativity. It's so exciting. We need more of it. Thank you, Man. And Greg, how about you? The lessons we should take with us today?
Well, I heard the word collaboration, and that's huge, both in terms of this vaccine and the research and COVID as well. But I also think it's important for all of us to not count out the dreamers. So many of the people behind the RSV breakthroughs and also the COVID vaccine were kind of dismissed. Their efforts were, maybe people were skeptical about them. And there's something to be said for giving dreamers a chance and allowing their persistence to finally pay off. I've become a little more patient with those who have these big, crazy kind of dreams, and we're trying to help people and try to save lives. So I applaud those dreamers in the scientific community.
Thanks, Greg. I would love to share that message with my children as they're thinking about dreaming in science and other fields. And how about you, Barney? What lessons should we take from your long work in this space?
Well, first, I just want to highlight the importance of basic science research and the importance of supporting basic research, because if anything, this story is a great story exemplifying how basic research on how the protein works, what it looks like, all with an eye toward human disease at some point, but really focused on basic mechanism, basic biology. This is the basis for how we got to this point. And then the concept that we learned in RSV was applied to the coronavirus problem. And so basic science builds on itself. And if I could be allowed to just say one more thing, that we're now looking into a future where we have technologies that can be effective, and we know how to use them. We know how to see things that we didn't used to be able to see that have changed everything. And we need to now look for the ways we're going to use that technology and deploy that technology and share that technology, because viruses don't have borders, and these vaccines need to be accessible to all people. And if we're going to take this technology forward, especially for pandemic preparedness and other disease concepts, it needs to be something that is being worked on in a global way. And so there's several other things we could say, but I'll stop there.
Yeah. Well, the importance of basic research is certainly something AAAS and science are very much behind, and creativity and collaboration, communication, the dreamers working in these spaces will help us prepare for the next pandemic. Thankfully, hopefully. That's a great message on which to end. We didn't get to all the questions today, but thank you for everyone who watched, and thank you again to our speakers. I hope you all have an excellent rest of the week, and we can look forward to seeing the results from the advisory committee as they weigh in on that second RSV vaccine any moment now. Thanks again, everyone. Have a great afternoon.
Okay, thank you.
See you all.